So we were able to select for a colony level trait the amount of pollen stored in the comb, which is a trait that no individual bee has. It's all as a consequence of the interactions of thousands of individuals, and we selected on that one trait. So we changed all those things. They go all the way back to the individual genes inside of these individuals who are interacting. We were successful in doing that. But along the way, we also constructed what I call a phenotypic architecture. A lot of things changed, and we then used the high and low pollen hoarding strains to figure out what else changed along the way. So first, let's ask the question, what is a phenotype? A phenotype is what you see, it's what you observe with respect to the individual or to the colony organism. You can have a colony level phenotype, the amount of pollen stored in the comb. You can have individual level phenotypes. Maybe the color of an individual worker honeybee. But the phenotypes are a consequence of a genotype and an environment. They both work together to give you the thing that you observe and you call a phenotype. A good example of that is looking here at flamingos. These individuals here, let's just say they all have the same set of genes in them, but this one here is pink. It's on the basis of what she ate. So the environment here is very important with respect to the phenotype. Over here, we can look at eye coloration, something that you observe in your own family and among your friends and people around you. And it has very little influence of the environment on it. It comes down to whether or not you inherit a dominant brown gene or two of the recessive blue genes. So you end up with brown or blue eyes on the basis of your genetic complement with very little effect of the environment. When you look at stature of human populations, you see that both things are working. So here are individuals from the Netherlands, Dutch people. They are much taller than individuals from Guatemala. And part of this is genetic, and part of this is environmental. You can look at the difference between these is a difference of 1914 to 2014. Did they change genetically, or did they change because their environment changed? Most likely it was the environment. Nutrition was better, less disease, and the stature increased. But the difference between the Dutch and the Guatemalans is genetic. They have genes that result in shorter stature. And there's still an effect of the environment with respect to looking at 1914 versus 2014. Phenotypic architecture, I look at it as like, making a balloon dog. If you take a balloon and you squeeze it, it'll pop out someplace else. Everything is connected to everything. Genes don't work in isolation of each other. So if you select for one trait, you're going to get other things that change along the way. Some of those things that change because they are affecting the trait that you're selecting. Some of them change just because when you squeeze the balloon, the air's got to go somewhere. They just happen because of the how traits are linked together. They're not in isolation of each other. Here's an example of phenotypes. Okay, so the example of the eyes. Of the eyes up here, those differences are primarily single gene traits with little influence of the environment, and it's just one trait, and you can see it. But down here, if you look at chickens, when you select and you breed chickens, you might breed them for color, you might breed them for the size of the wattles, you might breed them for their stature, but after you breed them, you have a whole architecture of the phenotype. And here's one with a long neck with very little feathers on it. And here's one that's very robust, etc. So there's lots of different things that go together here with respect to these traits. The same with dogs. Dogs, you've selected a whole collection of traits that distinguish the difference between, I suppose that's a Schnauzer and a Weihenheimer. And then also pigs. You get to see all these different phenotypes of pigs. And they've been selected for certain traits, but they have an architecture of those traits. So we studied architecture, the architecture of our high and low pollen hoarding strains, the social trait, by selecting for a single thing over all these generations. We only ever selected for the amount of pollen stored in the comb. But other things changed along the way. Things changed that enabled the increase or the decrease in the amount of stored pollen. To do this, we initiated what I call common garden experiments. So environment's important, so you need to control for the environment. So we took eggs from high-strain colonies laid by queens in high-strain colonies and eggs laid by queens in low-strain colonies. We took the eggs and we put them into a nursery. So the eggs and larvae being raised in the same environment. So we can reduce the effects of environment on the phenotype that we're going to be observing, which is going to be foraging behavior. We then took those bees, when they emerged as adults from the nursery, the frames were raised in the nursery colony, and we individually painted these bees. So we knew that they were either a low-strain bee or a high-strain bee based on the color we painted, and we put them into, again, a common wild-type colony. And what I mean there, wild-type, it's a colony that was not my high-strain bees, not my low-strain bees. It was neutral. So they're in the same environment at the same time. They're raised in the same environment, and now they're going to be expressing their foraging behavior in the same environment. We then collected the bees at the entrance of the colony once they initiated foraging, and we looked at their pollen loads, and we collected their nectar loads to see what the concentration of the nectar was that they were collecting and how heavy, or the volume of the nectar loads and how heavy the pollen loads were. If you take a worker honeybee and you restrain her in a tube such as shown here, and paint over her eyes with black paint so she can't see what you're doing, and then approach her with a single droplet of sugar water and allow her to touch it with her antennae, she will give a reflexive response. She will just automatically extend her proboscis. This is known as the proboscis extension reflex. She can't not do it. Providing the sugar concentration of that droplet of water is high enough. You can determine what her response threshold is to sugar by giving her different concentrations of sugar solution and see at which ones she will extend her proboscis. We also tested the bees for their responsiveness to sugar. So I showed you early on a little video clip of a bee responding to sugar that was being touched to her antenna. Her proboscis came out. Well, she responds to that sugar stimulus only if it exceeds her response threshold. So we took these bees and we tested them. We had a concentration series of sucrose that started with pure water and went up to 30%. So we had pure water, we had 0.1, 0.3, 1, 3, 10, and 30, a logarithmic series of increasing concentrations. We went down each of these bees individually, and we touched the antenna of these bees with the antennae, both of them, with one of these solutions. So we could go down and we could then say, well, this individual here responded at 0.3. This individual didn't respond until it was 10. Maybe this one responded at 0.1. So for each individual, we could determine what her response threshold to sugar was by observing the concentration of the sugar at which they would reflexively extend their proboscis. This is the proboscis extension reflex test that we use to test for their response threshold to sugar. When we did this, we found that the high-strain bees were more responsive to sugar. That means that they extended their proboscis to lower concentrations of sugar and even water. So they were more responsive to sugar than bees in the low strain. In the low strain, they had to have a higher concentration of sugar in the water before they would extend. Then when we looked at their behavior, we found out that the high-strain bees showed a pollen foraging bias. Most bees collect both pollen and nectar, but they can have a bias with respect to their loading. They load nectar into their crop when they're foraging, and they can get a certain amount in there. And and they load pollen onto their legs. So if you look at a bee that's biased for pollen, she'll have bigger pollen loads and smaller nectar loads, whereas one that's biased for nectar foraging will have more nectar and less pollen. So we measured that in all these bees, and we found that the high-strain bees had a pollen foraging bias. They also initiated foraging earlier in life. Remember the slide where I showed the age-related polyethism, the changes in behavior that occur? That transition to the onset of foraging takes place at a younger age in the high-strain bees than it does in the low-strain bees. When they collect nectar and water, they will collect lower quality nectar and water, meaning that their response thresholds to the nectar is they respond to lower concentrations of nectar and water, and they show up when they're foraging, that they will actually collect lower concentrations of these of nectar and also collect water when they're on foraging trips. And they're also more successful as foragers. Fewer of them come back empty. So when you're looking at the high-strain bees returning as foragers and the low-strain bees returning as forager, the high-strain bees will have fewer individuals carrying no load. And I ascribe that to they're more successful because they're more accepting. They have a lower response threshold, so they'll collect nectar from the flowers that the low-strain bees would just give up, go home, because the concentration was too low for them to really respond to. So then we wanted to ask the question, do we find the same relationships in wild-type bees? Now, these bees were selected, and they've got a phenotypic architecture, which I just showed you. That phenotypic architecture is associated with response thresholds to sugar and bias for pollen and nectar. But do we find these same relationships in bees that were not selected at colony-level selection for pollen hoarding? If you just take wild-type bees, do they show these same relationships? So we took wild-type bees and we paint marked them so that we would know the bees that we had marked. And these are newly emerged bees. So they're marked. We put them into a colony like this. This is a neutral test colony, common environment. After 4 days, we went out and we collected the ones that we had marked. We took them into the lab, and we tested their response thresholds to sugar, so that they emerged in there as adults, wild-type bees. We marked them. We put them into a host colony. We removed them at 4 days. We tested them for their response thresholds to sugar, and then we individually tagged them. We put little numbered colored discs on their backs. And so we could do something like, say, here's white 11. She had a response threshold of, say, 0.3 to sugar. And here's red 34. She was less responsive to sugar in that she didn't respond until it was, say, 30%. So we could determine what her response threshold was to sugar when she was 4 days old. Then we went and we collected the bees from the entrance as they were returning as foragers. We collected the bees that had numbered tags on them. And this is what we found. Here we have the sucrose response. This is their response to sugar. These are the different concentrations of the sugar that we tested. I told you before it was a logarithmic series. So down here, -1 was the lowest concentration of sugar. 1.5 would be the highest concentration of sugar. So individuals that didn't respond till up here, they would be less responsive to sugar than these down here. So if you took individuals that foraged on water, and then you looked at what was their average response threshold when they were 4 days old, they're returning with water, what you found was that their response threshold to sugar was very low. In other words, they were very responsive. So low response threshold means higher responsiveness. So they're the most responsive. Then the pollen foragers, they were the next most responsive. Then individuals that collected both pollen and nectar, that collected nectar, they were next. And then those that collected both, and then those that returned empty. The ones that were empty were the least responsive to sugar. So you can see a direct relationship here of the behavior, the foraging behavior, to their responsiveness to sugar when we tested them at 4 days old. Also, nectar concentration, the concentration of the nectar they collected correlates negatively with their response threshold. So if they have a higher response threshold, then they would return with only higher concentrations of nectar. If they have low response thresholds, they're more accepting, and they'll return with lower sugar concentrated nectar.